Welcome everyone to the CIBM Center for Biomedical Imaging Breakfast and Science Seminar, number 14 we are now. And it's my great honor and pleasure to introduce Mary Bach, who actually is the initiator of the Breakfast and Science, and which dates back to already 10 years ago. So in 2011, she started it. And I would like to just give the floor to her and introduce our speaker for today. Thank you, Mary.
Thank you, Pina. So welcome everyone, and today we are very pleased to have with us Professor Cristina Granzira. And so I think most of you know her, but I've prepared an introduction for those that doesn't know her, because she has been around since many years now. So Cristina, she's a MD from the University of Padova Medical School, and she has a PhD in neuroscience from the University of Lausanne. She obtained the Swiss Neurology Board in 2010. And in between 2011 and 2014, she was a lecturer and senior lecturer in the University of Lausanne. Then she moved to the MGH, the Massachusetts General Hospital, and the Harvard Medical School, and the Harvard Medical School in Boston. In there, she was assistant professor in radiology and assistant in biomedical engineering. And finally, luckily, she came back to Switzerland in 2018 as a leading attending neurologist in Basel University Hospital as SNF professor at the Biomedical Engineering University of Basel. And in that is where she created this translational imaging in neurology, the theme group that I think she takes some time to present as well. And she's leading a multidisciplinary team of 36 people. So MDs, radiologists, neurologists, engineers, PhD students, and postdocs. So she's doing a lot of things. I'm sure that we will see it right now. I would like just to highlight three projects. The first one is the Insider, the imaging axonal damage repair in multiple sclerosis, where she studies microstructural MR imaging biomarkers. She's as well exploring atrophy in the Swiss multiple sclerosis cohort, atrophy of the brain, of the spinal cord, and how these affects, the atrophy is affected by the treatment and repair. And finally, she's as well working a lot on multicentric large-scale studies for decision support with very advanced biomarkers like the central vein cortical lesions or the paramagnetic rim lesions. I'm sure she will present this. So overall, her aim is to model the disease and impact, disease impact and evolution, and how we can integrate this multimodal MRI framework to do so. And to do all these things, of course, she got a lot of grants. So she has awarded by Ambizione and the SNF professorship. She got as well NIH grants, and she was awarded with the prestigious Translational Research Award from the University of Lausanne and the Innovation Award from the AGH. And on the top of this, she's doing other activities. She is currently the co-president of the Medical Scientific Advisory Board of the Swiss MS Society. She's junior faculty representative of the Neuroimmunology at the European Academy of Neurology. She's member of MAGNIMS, the European Academic Network of the MRI in MS. And she's a scientific advisor in many boards and scientific societies and healthcare companies. On the top of this, she's able to answer the emails super quickly. So the point I was checking when I met you, Cristina, and I don't remember. Usually I remember when I met people. And I was checking the emails. So this is a strategy that Jean-Philippe teach me. And the first email we have is from June 2nd, 2008, where you were already inquiring about the PACS research that we had at the CIBM at the time. And then the next mail I have from you is something about Feurenzangenbowle. I think it was a Christmas party German thing or something like that. I was invited. So this was our first exchanges. And of course, more seriously, since then we had the opportunity to work in projects common, like the development of the work in progress package Morphobox at the time of Siemens, We were as well working together already in a stroke with Alessandro. I remember that. And we were as well exploring with Gunnar and you the MP2 RAGE tissue segmentation. I don't know if you remember. What was the inflection point with my collaboration with Cristina was in November 2012. Well, I got a call, so I was in the office at the time in the hospital, and she called me because she got spontaneous candidacy of a student from a Swiss Excellence Fellowship. And she told me, Mary, I want you to co-supervise as a student with me, be confident in me, because you got a very good impression of this student. So this student was Mario Joao Fartaria, and she was not wrong. He was a great student, and he actually got one of the best FBM PhD thesis in 2018, I think. And I always thank her for the confidence she had in me and to involve me. So since then, I've enjoyed really a lot co-supervising and doing this research with you. It's very funny to have meetings with her, scientific meetings. She has a concentration of ideas per second impressive. So I had time in following her. And she's doing that in movement, let's say. So she's often not still. I'm surprised that today you are still in a desk, because usually she's either catching a train, maybe you can cut this pinna, sometimes driving on the phone. So it's really amazing. What I admire the most of her is as well the fact that you understood she's expert in her field, but she's expert as well in other fields. So she is expert in MRI, even in MRI physics, I would say, and she's as well very, very knowledge in image processing. So she has really very good ideas as well on how to improve the methods. And I wanted her to present as well today, because I think she will present it, because one of the most important things is this translational aspect. So you see the name of her group. And this is, I think, as well, a common aim that we have at CIBM to make these tools available. And for her, the most important thing is how this can help at the very end, for instance, taking the decision to change the medication of a patient. So I really admire this from her. What else? Yeah, so something I want to as well say is that she's sharing as well her network. So thanks to her, for instance, we could enlarge a lot our collaborations with people from NIH, now with people in Belgium. And I think this is very valuable, and I really like it. And I hope, Cristina, that we will be able to collaborate in the future with new projects. So you understood that she's for me a reference as a researcher, for sure, as a group leader. And I would like to thank you to accept my invitation today. And the floor is yours.
Thank you, Mary. After such an introduction, I have not too much to say. No, I mean, I think it's clear the summary of all this, which is way too much. I told Mary to say 3 lines and she wrote 2 pages. The summary of all this is that I'm actually a weird MD, a weird neurologist who really likes working with physicists, engineers, computer scientists, and also has a lot of friends in this community. So most of the people today, present today, are and were my friends. So having said that, let's cut it short unless I become all reddish, and let's start with the presentation. I mean, I normally like the interactive presentation, so I don't like lecturing at all, and it will not be a lecture, it will just be a sharing of some projects, some experiences we have done by using advanced MRI applied to multiple sclerosis patients. It will not be an exhaustive presentation of the work we have done over the years, because there are too many, too exciting, and every time I'm trying to cut down on slides, I add more. So I'm not very successful in this. That's the outline of what I would like to present. I would like to first introduce the people who are really doing the work in collaboration, and you will see how many collaborations we have, especially with Lausanne. It's Think Basel, the group, so translational imaging in neurology. Then we will make a very brief introduction about the pathology that we will try to better understand today, which is multiple sclerosis. I will focus on how we could use, or we can use, or we have used advanced MRI to understand the MS and perhaps also to better care of MS patients. And then at the very last, I would like to show you some results of the collaboration, especially with Mary, which has been over the years very successful, as you saw. So we had also a lot of parties together, not only the Feuer Zangebol at the beginning, but these parties were also times to exchange. I mean, ideas are not coming always in meetings, in formal settings. They're coming by doing things together. And that's why I think most of my closest collaborators are also friends. So now let's start with the people who are really doing the work. So I think Basel is a big group. We are actually, I counted them by doing these presentations or preparing the slides. We actually 36 now, and we are organized in teams. So there are, I think, Basel 5 teams led by senior researchers with students. And this is in the center, my team. I have the honor to coordinate and head this Think Basel group, which is multidisciplinary efforts. There are medical doctors there, there are engineers, there are physicists, there are computer scientists, and we try to understand each other, first of all, and then to work together. And Think Basel is in neurology in Basel University Hospital. It's also in biomedical engineering. And recently, the former chief of neurology, Professor Kapus, that you see here, who's one of the most well-known MS experts in the world, founded a new institute, and Think Basel got also the leading part of the imaging in this new Neuroimmunology Institute. So having said that, I wanted to show you what the team I'm working with is taking care of. So as Mary said, we are mainly working with advanced MRI techniques, not only because we are also responsible of the analysis of the Swiss MS cohort, but what we are mainly doing, and if I can move this here, is that we are trying to understand and combine and use advanced quantitative MRI together with clinical measures, with genetic measures, with fluid biomarkers to better understand. That's essentially better understand multiple sclerosis. This is our main goal, but not only. We have also projects applied to neuro-HIV patients, to cerebrovascular disorders, and so on. And what we do is we are structuring, so we are actually having 3 pillars activity. We have many clinical translational studies. I counted up to now, not only from my team, but from the whole thing. There are 7 ongoing studies. 4 of them are led by the insider team. We have post-mortem studies, and you will see how we use post-mortem MRI to understand what we find in vivo, but also to trigger new ideas and new paths of exploration in vivo in patients. In the end, we care about what we find in patients because we have the hope that all this information can be translated in our practice. And last but not least, this is also part of the collaborative work. We are evaluating new contrasts together with the medical physics department, also with the Siemens team in Lausanne at a certain time. And we are validating new analysis tools that have been developed by different teams, especially Mary's teams. We have a flow back and forth of information. So we provide manual segmentation, we provide feedbacks, Mary is providing improved tools. The same thing happens, for example, with Alessandro Daducci in Verona. We have this bilateral workflow, which allow us to validate and make step forward, not only the development, but also in the translation into the clinical practice. Having said that, now I would like to show you some of the works that we are currently doing. And we are mainly dealing with multiple sclerosis, which we perhaps know already is an autoimmune, chronic, inflammatory and degenerative disease of the central nervous system, which unfortunately lasts for the entire life. It's not a disease that you can cure. It's unfortunately a disease that affects young people, more than 2 million actually worldwide. And it's a disease that you can, however, treat. You can treat inflammation. To a certain extent, you can also limit neurodegeneration. And what we actually are fighting against are essentially two types of inflammatory and degenerative events. One is focal, we have lesions in these patients, and one is diffuse. We have diffuse inflammatory activity, but also subtle degeneration. Then in the end, leads to a loss of structure, loss of brain and loss of spinal cord structure. And all this damage is essentially due to lymphocytes that are auto-aggressive. They are attacking a certain part of our central nervous system, which is the myelin sheath, here in light blue, surrounding the axons of the neurons. The nice thing is that we are well constructed, I always tell the medical students, because if there is a damage, our central nervous system tries to react. So there is a spontaneous repair activity and then damage again. So it's a wave that repeats and it's not regular. In fact, the clinical presentation of these patients is essentially a mess. We can have everything. We can have patients that have just damage and no clinical symptoms or signs or deficits, and those are the RRS patients. We have patients that show essentially inflammatory signs and symptoms, and there are other patients that show essentially neurodegenerative processes. We see a slow progression of the disease, a slow increasing in deficits over time, and all these phenomenon. So here we tried, of course, we are medical doctors, like physicists, we like categories. We try to put tags, we try to understand the disease by categorizing, but it's most probably, this slide is most probably not appropriate. This is a continuum of inflammation, neurodegeneration that is intermingled and not very regular. So it's a big chaos. And sorry, I wanted to go further. And what we know from the clinic, this is what we are currently doing in the clinic. We know that this disease can be visualized structurally. We have lesions in the brains that we can see in T2 weighted scans, typically indicating a lot of phenomena. These T2 lesions are inflamed lesions, so with edema. There are also lesions which are repaired efficiently or not efficiently, we don't know. They're all white. On the right side of this slide, you see another type of image which we use in the clinic, which is a gadolinium enhanced T1 image. And this is an image where you can see the breaking here. I don't know if you can see my mouse, but you see the breaking up of the blood-brain barrier. So there it's an area that we can visualize where active inflammation occurs. But of course, GADO is pretty big, it's 300, 400 kilodaltons. So there you see only the big breaking of the blood-brain barrier. And if we go on, in the clinic, we have also T1 weighted images that allow us to see when there is a lot of neurodegeneration. If there is a hole here, like the one you see here, the hole is represented by a black spot in this T1 weighted image. And when there is a lot of neurodegeneration leading to volume loss, we can see it and describe it qualitatively, saying there is atrophy in this brain. So that's the final, that's everything we can say so far in the clinic. And now I wanted to step into the central part of this, let's say, lecture or presentation, call it as you want. For me, it's mainly a discussion about what we have found so far. And we have mainly applied in these studies that I'm going to show you later on, what I call, or what is called in the community, quantitative MRI. And here it's a recent review that we have prepared together with the Magnums Consortium about quantitative MRI in MS. And you can see how many things we can do. We can have, through quantitative MRI, we can gain information about how these white dots in the clinical images are so different. There are different types of images, of lesions. There are lesions which are more or less inflammatory. There are lesions which are more or less damaged. There are lesions which you will see in the next slides are linked to some histopathological types where the innate immune system is active. Those are called chronic inflammatory lesions, and so on and so on. And quantitative MRI has helped us to understand that we are not dealing only with one white lesion. We are dealing with many, many subtypes that most probably have different implications for our patients. We are also, through quantitative MRI, we were also able to study outside the lesions. So what's happening in the normal, I said before, that the normal appearing tissue, there is a diffuse inflammation and degeneration there that is not visible in our conventional images. And the interesting part for us is that there are techniques in these quantitative MRI fields that are sensitive, but also specific to a certain extent to the myelin sheath surrounding the axon. So this is a chance for us to study the remyelination, the repairing, the efficient repairing process. In fact, you see, when you have an MS patient, it's typical of an MS patient who's presenting with a relapse. He has, I don't know, a decrease in visual acuity, for example, but after 1 week, 10 days, the patient is recovering. And this is due to the resolving inflammation, but also to the remyelination process, which starts and occurs attempting at repairing the damage. And let's start with the studies. I mean, this is something I could not show you, but I do. It's actually the proof of another paper that we very recently published, and where we have combined myelin and axonal sensitive surrogate measures to study in vivo in MS patients what's happening in these white lesions that you saw in the FLAIR image before. And to do this, Reza, who's the first author of this, is a PhD student in our group, studied 91 MS patients and 72 healthy controls. The patients were slightly older than the healthy controls, and they had a disability range, which was very broad. I mean, patients with 0 disability have nothing, you cannot see any deficits. 8 disability, those are patients in wheelchair. Those patients had different treatments, and 62% of them were in this inflammatory phase where we see clinically the inflammation. 29% didn't have any clinically evident inflammation, and they were the so-called progressive MS patients where degeneration dominates. And here, what did we do? We used, in these patients, we of course acquired conventional images like the 3D FLAIR. We used MP2RAGE, which is a sequence developed in Lausanne, and that now has reached the level of being product and being distributed everywhere. So MP2RAGE we use to look at the damage in the cortex. You see how well you can see lesions in the cortex, or here, the little one, or touching the cortex, the so-called leukocortical lesion. So we use these 2 images to identify the presence of the lesion. And then we used other quantitative maps to understand better the structure, the heterogeneity of those lesions. So look at those 3 images, for example. This is a myelin water fraction map, and this is a neurite density index map. And now I'm trying to, in 2 words, to tell you what we gain in terms of information from these 2 maps. So here we see a white lesion, totally white, perhaps a little bit heterogeneous. Now I have German words that are coming in my speech. But what is impressive, I think, is that you see the myelin water fraction map showing a reduction of this myelin water fraction, which is pretty localized on one part of the lesion, and the NDI, which is showing a reduction of this parameter, which is very diffuse, actually more concentrated in the center. This tells us already, without knowing what these 2 maps mean, that there is something going on which is not homogeneous. This myelin water fraction map, if I may say conceptually, it's actually a measure which is directly related to the properties of the myelin sheath. So what this sequence does, and other sequences doing myelin water imaging, essentially they capture the information from the water which is trapped between the myelin sheaths. So this is a special water because being trapped there has a special relaxation time, which is very short, and you need sequences that are capturing the signal produced by the relaxation of these water molecules. And this is one of the sequences that can do it. And then what we have is a map that gives us the fraction of this pool of water molecules. Here on the right side, you have another measure, which is a measure of intracellular water. And this is derived from the modeling of the diffusion signal in a certain diffusion experiment. This is a multi-shell diffusion experiment. And it's actually one of the variations of the so-called standard model of this diffusion signal. So it's nothing very fancy, but it's an interesting measure, which give us, again, this water content in the intracellular compartment, which typically in white matter is related to the axonal compartment. Then we will see later on that it's not a very specific measure. So I consider them as surrogate measures, or the community consider them as surrogate measures of myelin content and, let's say, axonal related content. Then what we did here, we applied directly the tools developed by Francesco and Mary in Mary's lab to detect automatically these lesions and the cortical lesions. And then, of course, we did the manual correction of this automatic, giving feedbacks to Mary. So you see already the interaction in a totally different study. This is not finalized at the development, but while using this tool, we give feedbacks and we get back also improvement in this. Right. So now we looked at different type of lesions by using these two maps and trying to disentangle how the behavior of these lesions is. So first of all, we looked at white matter lesions on the whole, and we saw, of course, that in the lesions, the white matter, in the white matter lesions, the myelin water fraction and this NDI fraction was not fraction, this NDI average was decreased compared to normal appearing tissue and healthy tissue. This was totally expected. The same we observed in cortical lesions. So these are things which we proved correct. And we said, okay, if the things are like this, we can go on and look at different subtypes of lesions, which we did. We looked actually at periventricular lesions and juxtacortical lesions. Periventricular lesions are thought or were reported in post-mortem studies to be particularly destructive. Whereas juxtacortical lesions, because of the anatomy of this juxtacortical area, which is fascinating, there are different oligodendrocytes, different ratio of oligodendrocytes compared to the bundles. There is also more iron in there. Anyway, this region is particularly prone to remyelinate and repair. So let's say from the neuropathological studies, we supposed that there was much more damage in the periventricular compared to the juxtacortical lesions. And this is exactly what we could find by looking at the average of these two parameters in the two groups. And the same hold it true for other two types of lesions, the so-called rim plus lesions. Those are lesions which show in QSM images a rim of paramagnetism. And you will see later on, I have a lot of images to show you what I'm speaking about. So these lesions are actually extremely interesting because they have a rim of iron, which is most probably localized in microglia cells, in macrophages, which are activated, producing a lot of toxic substances, and they're very destructive. So we know that those lesions harbor chronic inflammation, smoldering activity, which we are so far not able to treat, and they're more destructive than the others. And actually, by using this averaging of myelin water fraction and NDI, we saw exactly what we expected, that these rim lesions were more destructive than the others. And then we asked ourselves, hey, do the patients who are progressing a lot, where we don't have too much inflammation, show more damage in the lesions than the patients who are actually a bit younger and with a lot of inflammation, not too much degeneration? The answer is no. Lesions are the same or seem to be very similar among these two different clinical subtypes of MS, this relapsing, remitting, and progressive MS, telling us, once again, I already told you before, that these subtypes hold, they have little value. And then this is something that was also very interesting, of course, for the diffusion community, NDI, which is, we performed the analysis of NDI using two different standard models. One was not and the other one was MCBMI. obtaining the same result. But of course, MDI is obtained with these models is a pretty controversial measure. We use it as a surrogate marker of intra-axonal or intracellular compartment, which in white matter is the intra-axonal compartment. But of course, there are studies showing that this NTI is not so much related to axon. It's also related to myelin, and even more than the axon. Then there is the other part of the diffusion community of saying, no, diffusion is not sensitive to myelin. So there is a big debate there, which is at the time, let's say, an intellectual debate or a theoretical debate. On the other hand, it's a debate based on data. And this Grusso study showed actually that in the spinal cord, this parameter was highly related to myelin content and much lower. There was a much lower relationship with axonal content. Then, of course, there are people who are saying and demonstrating that these multi-shot diffusion experiments are TE dependent, which means that if you have an acquisition that is performed in a certain setting with a certain TE, you're more or less sensitive to the myelin. And of course, we didn't take this into account because in a clinical experiment, it's impossible at the present time or in the current condition to perform such an experiment where you have multiple TEs and you see. So this was not possible. What we could do using this study was to model. And what we did, we modeled statistically the contribution of these two parameters, this NDI, so the intraxonal, and myelin water fraction, to separate the tissues. So the white matter lesions from the normal appearing tissue and from the white matter tissue in healthy controls. And what we saw actually is that, the animations were wrong, but is that for white matter, this NDI parameter contributed more than myelin water fraction to the separations of these tissues, statistically speaking, telling us that from a statistical point of view, NDI is providing additional information compared to myelin water fraction. Of course, you can discuss about the different characteristics of these sequences. We're not going into that discussion, but this is one approach to not solve, but to try to deal with the problem of accuracy of these models in the real practice or in the application of the models to a certain clinical question. And then I'm open to a lot of questions about this because this is something which I'm really fascinated by. Last but not least, we looked at the same parameters in the normal appearing tissue. Also there we were astonished to see that in the normal appearing white matter, there is a huge amount of damage quantified by these two parameters. Here you see the comparison between MS patients and healthy controls for NDI and myelin water fraction. And even more astonishing was in the normal appearing gray matter, because there was not only a difference between MS patients and healthy controls, but also between these two clinical subtypes of patients, telling us that something is going on in the cortex, as the neuropathologists already told us. That perhaps explains why one type of patients is progressing and the other not, or contributes to the explanation. Then I wanted to show you another couple of studies that we're doing using the same approach, using different type of quantitative parameters. This is a study from Pojulu, another PhD student in our team. Poju is a deep learning person, and he really likes classification. He really likes the algorithmic part. And of course, because of this, he has been working together with this artificial intelligence team of Siemens in Princeton. He has also collaborated with Mary. Mary is involved in everything I'm telling you about. So what did Poju do? So here his question was, hey, we have so many contrasts in this study. Can we understand what is the most important one or the couple of most important contrasts? They all provide us a complementary information about what you want to know, Cristina. So they always tell me, I want to know. And they try to find ways of understanding better. And what he did, he actually made a new approach. So he actually developed this gamer MRI, which is based on gated attention mechanism. And he performed a ranking of this multi-contrast quantitative MRI to answer certain questions. He actually developed this in stroke patients. And then he applied it to our two different types of lesions, this periventricular juxtacortical, and he came up with a ranking which made sense to me from the pathological point of view. So he understood that among these four contrasts, quantitative one and multi-water fraction were contributing most to the differentiation of this very different type of mass lesions. And the same thing then he did with the multiple diffusion parameters. We are overflown with diffusion models and models of microstructure, all providing different type of information, but also very similar, bit complementary. They are all telling us something which may be potentially interesting. And Koju said, let me check. Let me see what is the one that for our purpose could best answer the question. So he checked using MRI, he assessed which type of parameters were most sensitive to differential lesions from the tissue around the lesions, the so-called perilesional tissue. And he came up with a couple of parameters that then he merged in one map. And then this map was validated by relating it to the clinic of the patient and to biological marker of the neurodegeneration. And interestingly enough, he found actually correlations that were, for me, as a clinical or biological person, amazing. Through a combination of parameters. This is not something you see normally in the clinic. You see correlations much lower, and those correlations were overperforming the one of the single parameters, telling me again that there is complementary information there we should be able to exploit in a clinical application. Then as a next study, if you're still bearing with me, because I have a lot to show you, this is actually everything you saw so far were published studies. This is something I wanted to share with you, is a study which we have just submitted that has been published in abstract forms in Trans and INSRAM. But in this study, we did something different. So you know this slide I showed you before. What Reza did again, this is wrong. Yeah, well, this is the same slide before. That's why it was put like this. But anyway, this is Reza's work. Reza told me at a certain point, I like these QSM images. And I said, what do you like? It's a little bit like this. I didn't really understand why I like them so much. And then he explained me, hey, I see different things there. I don't see only these rim lesions that you see here. I see a lot of types of lesions. And then he came to me and he told me, I see 5 types of lesions. And I said, cannot be. I mean, my answer is often cannot be, because it has not been described in literature, because, I mean, there are other people who are doing the same stuff. Why should you see more? That's always my answer. Then he tells me, hey, no, I've looked at least more than 2,000 lesions, at almost 3,000 lesions. A lot he discarded because there were artifacts, because QSM was not so nice. Anyway, and he tells me, I see lesions that are invisible. And I said, how can you see invisible lesions? And he tells me, hey, we use the mask from Mary's and Francesco's algorithm. I just registered it to QSM, different with a certain registration algorithm, and I don't see a lesion in QSM. That's the first thing, which has been described, but not explained. Then he tells me, I see black lesions. I see hypointense lesions. This has never been described. And then I wanted to see the examples. And the examples is here. You see this little black. Well, it's not black, it's a little bit grayish, but it's hypointense. Then he sees hyperintense lesions. There we were fine, because those are the lesions that have been described a lot. Then he tells me I see hyporim lesions, so lesions with a black rim around. You see it here? I mean, I'm used to this. It's not super evident, but it is there. And then he sees the classical rim lesions, the bad ones that I told you before. So one has been described, not very well known what it is. This has been described. People try to understand what it is. This we understand. These two, we don't know what they are. And then he makes an estimation of the prevalence in this population of patients that we have for this different type of patient. And then, of course, what do I tell him? Give me something more, because I don't know what they mean, these lesions. And so what he did, he actually evaluated, of course, this is a qualitative classification of those lesions. He quantified the susceptibility in QSM lesions, in the QSM subtypes, and we got, of course, what we expected. I mean, we selected the lesions according to their susceptibility appearance. So this is nothing fancy. What is more fancy is this. Because then, of course, Reza, who did the previous work with Mile water fraction and neural density index, told me, hey, now I quantified these two parameters in these lesions, and look at that. So this is actually the average for each patient of this lesion type. And you see that there is actually a certain trend and a difference with the lesions, which are these rim lesions. No, these rim plus lesions are, let's call them the bad ones. They're the most destructive one. They're actually on this part of the graph. So they show the lowest parameter on average than the others. And then you have these two lesions, which were very interesting for me, the ISO and the hypointense, which have shown a parameter which is very similar here. It's very similar to the normal appearing white matter. And here it's not, let's say, the nearest to the normal appearing white matter. And then, of course, I still didn't believe him. And we were discussing also with Danny Wright at NIH, and he was saying, this may be perhaps very hot, but you need to show us more. So we decided together that Reza had to do some modeling and some clustering of these lesions. So what Reza came up with actually is very good, because he's always following and doing what we suggest. Sometimes it's a little bit challenging to fulfill. But anyway, so what he did, he took these lesions and he tried to cluster them. He clustered them according to these two parameters, NDI and Wally water fraction, and their susceptibility. And he found actually two clusters. And the interesting thing was that in the cluster with the lowest NDI and Wally water fraction, we had 80% of these bad lesions, which here are called rim plus lesions. They also called PRL lesions. Then we modified this graph for a paper to render the terminology homogeneous. But anyway, it doesn't matter. These bad lesions are in the lowest part, almost all of them. And these two other very interesting lesion types, these hypointense and isointense, are in the good part of the graph, so in the good part of the cluster. So then what we do, we went post-mortem. Post-mortem, thanks to Matthias, and you will see later on his efforts, Matthias Weigel, we could have the same protocol, quantitative MRI protocol, acquired post-mortem. And you will see how we do the post-mortem registration, acquisition, and blah, blah, blah. But here, what I wanted to show you is that we try to understand how these lesion types are looking like post-mortem. So we know that in histopathology, there are some lesions that are totally uninteresting. They're called chronic inactive because there's nothing going on. Here is a myelin staining. You see myelin is totally destructive. It's destroyed. There's no myelin. And sometimes there is iron in these little cells there, the microglia cells that are stained in blue. Sometimes there's not. But anyway, they're not very active, these lesions, so they don't do anything. They're just destructive. And these lesions post-mortem look like very hyperintense. Then we looked at the lesions that we know, these rim lesions. Now we know that those are lesions very destructive. They still have this iron-rich, you see here, microglia at the border, here at the border. And these lesions typically show this rim of hyperintensity, and inside it's a little bit darker. This is a specific pattern of these lesions. Here you see also little rim. So this is all something that we could suppose for these lesions, and we knew for that. So nothing new. The interesting part comes with the remyelinated lesions. The remyelinated lesions post-mortem, they look like this. They're very different. They're lesions which are completely remyelinated, this and this. Here we still have some microglia cells, you see, inside. They are not rich in iron, but remyelination is a bit incomplete in some parts and very complete in others. And look at the QSM. So in the complete remyelinated area, QSM is isointense. Whereas if remyelination still has to happen or is still ongoing here, or is not complete, here's a little hyperintensity in QSM. This one, which is below the cortex, very interesting location. Below the, I already said it, I repeat myself, but I'm really convinced. This one is fully remyelinated. There's nothing going on. I mean, they already did the microglia cells of their work of remyelination, and the oligodendrocytes as well. We don't have iron in these microglia cells. You see also there are no actively remyelinating cells. This is the biggest staining, and it's hyperintense. We had a couple of those lesions, so we didn't have a large numbers, but we have 15 or 16 lesions in this study. However, we saw also different things. We saw that remyelination can be quite complete. We have still some iron in microglia cells, and these areas in QSM are isointense, but they still harbor a little bit of hyperintensity. So there could be mixed phenomena going on there. And there are also remyelinated lesions, which are not completely remyelinated. So it's a partial remyelination, still harboring iron-rich cells, which look totally hyperintense. So our conclusion there was that if we see an hyperintense or an isointense lesion, this is for sure something where remyelination occurred, for sure. This, we have data supporting the evidence that remyelination occurred. From the physics, you also understand this, why you don't understand very well the hyperintense, but I'm also open to questions there, and we have some theories to explain it. You explain the isointense, and from the signal, it's also understandable that if there is an incomplete remyelination, this QSM is still a bit hyperintense because it's very sensitive to both iron and myelin orientation. And if there is iron occurs, there is an even higher hyperintensity. So there is a big spectrum of remyelinated lesions that we are going to investigate in future studies. And also something really fascinating, because I'm speaking a lot, I'm now preparing this microstructural plenary for ISMREP together with very good people, and I have the chance to have before me in the line Yonko Lee. Yonko Lee is an expert in QSM, a young guy, Korean guy, super active, who not by chance, he has been working for years on that, but he is working on a model to disentangle the effect of iron and myelin in QSM images. And this was bingo for me. When I heard this, because I listened to his presentation, in preparation of this ISMREM, I said, hey, Yonko, can we merge efforts? And that's what we're doing now. So we're using this model. Don't ask me to explain you the model, I will not, but you listen to Yonko's presentation at ISMREM. So he has actually developed a model that now is under evaluation, already out in Biotics. But this model now has been applied to our postmortem data. This is what he gets from a QSM image in terms of myelin content. You see the capsule interna here, very rich in myelin. This is the corpus callosum, very rich in myelin. And this is the relative iron map that he obtains from the same QSM map. So we are totally excited to see what's coming out now that we can disentangle iron and myelin in these postmortem images and then correlate them with our histopathological analysis. And now, if you bear, I have still a lot of stuff. So you need to take a coffee, drink a coffee, and continue listening, because it's getting a bit nearer to the clinic now. So as I said, this QSM at the beginning didn't convince me, but now convinced me a lot. And these images, these susceptibility images, they're also very rich in information, even if you don't model them using QSM. This is an example of images that you obtain using a certain susceptibility experiment. It's a 3D EPI from the NIH group. And using this approach, the NIH group was pioneer in understanding specific lesion subtypes. So what did they do? They use actually these phase images, which are normally crappy, but if you unwrap them, they're a little bit more decent. And somehow they give us a lot of information, which was neglected for a long while. So in these phase images, they showed them that some lesions have this famous rim that I told you about. And this famous rim, you can look at it in phase images, you can look at it also in QSM images. And they use the phase. And what they discovered, you see how beautiful it is. And this is actually a rim of activated microglia and macrophages, which are perpetuating inflammation at the border of those lesions. And they showed through a post-mortem and in vivo study. So, in vivo and post-mortem, they had actually the chance that they followed up a patient for many years and then he passed away. So they did this. They also did independent post-mortem MRI studies. And they showed that we can see, using this approach, almost half of the lesions that have those characteristics post-mortem, which is already a lot. And they also understood that these lesions are somehow not static. They're increasing slowly on average over time. So they're somehow not only having inflammatory activity, but doing also something. They're increasing. They're doing some destruction around. And interesting for us, and the most interesting part is that the presence of these lesions is associated with decreased disability or dysplasias. Everything is clear. I think we have understood everything, but okay, now we need to understand what does this mean for my patient. So does it mean that if I have these lesions, I'm necessarily going to progress? Or is it just an average study? What is the single subject telling? And here to do that, we did to plan this study. This is a study, again, in collaboration with Lausanne. It was driven by, well, together with Pietro Maggi, who was in neurology in Lausanne, again, together with Mary. There is a lot of activity going on between Basel and Lausanne. I promised it when I left from Lausanne, and I think I'm fulfilling the promise now. So this study is a collaborative study between Lausanne and Basel, where we asked ourselves the question, okay, we know these lesions are bad. They are inflammatory. We know that we can quantify in MS patients biomarkers of neurodegeneration in the serum. Are those lesions linked to these biomarkers? So if I have those lesions, can I see that there is increased neurodegeneration in the serum of those patients? Because this can lead me to a combined biomarker potentially in the single patients. We know that this is the case for gadolinium enhancing lesions. So when you have activity, let's say acute activity in these patients, this biomarker is largely increased. So as we have this with acute activity, do we have it also with the chronic activity? And this is our question. And the question was answered applying this susceptibility MRI to patients from Lausanne and Basel. We acquired actually the same susceptibility-based MRI in 118 patients that are organized here. And then we divided the patients in patients who had more than 2 of these lesions. I mean, we checked the distribution of data and we said, hey, there's a group which has more than 2 and a group which has 0 to 1. And these groups were actually different. This is a univariate analysis. It has the value studied as, but what we found there already was that this group with more equal 2 of these rim lesions had much higher SNFL. Of course, we know that this SNFL, which is this biomarker of neurodegeneration, is influenced by a lot of factors. The first factors, these acute lesions, we took it away because we chose patients who did not have any of those lesions and who did not have any relapse. So in the last 6 months. So we had the cohort of patients which was purely non-active, acutely non-active. So this is one factor eliminated to understand if these rim lesions were associated with an increase of this biomarker. And then we did complicated statistics again, and we include in this model all the factors that are known to influence this biomarker. And we assess the contribution of these factors to the biomarker increase. And if you look at this graph, you see that the presence of more than equal to PRL is the factor which is mostly influencing the increase of this biomarker. Even the treatment doesn't influence it or the disability of the patient. Here, actually, what we did, we use an SNFL percentile. So already SNFL, which were matched for age with a large cohort of, I don't know, I think it was 10,000 healthy controls. So the effect of age, for example, was not considered in this model because we had already percentiles of SNFLs. This was amazing for us. And where we want to go now is the use of this advanced biomarker in the clinic together with SNFL. So what you see here is a patient with here, this one had 8 of these type of lesions, these rim lesions. It has skyrocket high SNFs here. This one with 0 had very low SNFs. So a very interesting potential combined biomarker of chronic inflammatory activity. And why am I so excited about this? Because this is something that we're not sure we are treating. So with all we have, I don't know how many treatments in MS now to slow down the progression, but we are mainly targeting this acute inflammation. We don't know if any of those compounds are really active towards this chronic inflammatory activity. Those are different cells. And this is the fascinating part and where we think we can make a contribution. At last, of course, going back to the pathology, we also did the same analysis in the pathology samples that we had the chance to analyze. We saw that these rim lesions were associated with increased, not only increased microglia here on the border, we knew it already, but also increased axonal degeneration at the border and also in the center. And this is a further demonstration that there is a pathological link of this in vivo finding that I showed you before. And that's a quantification of that. So you see that the 2 types of chronic inflammatory lesions that are associated with this rim, they are characterized by a high level of axonal damage. This is acute axonal damage actually at the border. And last, I would like to show you the post-mortem experience. This is not even a paper yet. This is a talk at ISMRM. The program number, you see it here, so I do a bit of publicity. It's a huge collaborative work again. It's a work of Riccardo Galbuzera. He is a neurologist who somehow wanted to do a PhD. Don't ask me why, but he's doing a PhD. And I have nothing against the PhD, but somehow it's weird that first you do neurology and then you do a PhD because you need to be really motivated. So anyway, Riccardo is extremely motivated. And what we are doing is this. So for this work, we are collaborating with Göttingen in Germany. Göttingen, why? Because the chief of the neuropathology department there is one of the most worldwide known pathologists in MS, because they are heading also the German biobank for MS brains. So there it's an easy setting, and they have the same scanner. So they have the same Prisma that we use in Basel. And somehow Matthias, who is our main senior physicist, actually the only one, but we take a lot of care of him, he actually is traveling often to Göttingen to set up protocols, to improve protocols, so that we have a compound of comparison between in vivo and post-mortem. Here you see how we do the things. I mean, a lot of people in CIBM are doing post-mortem work, many in animals, so it's nothing new perhaps from you, but the fancy stuff is that. So we use actually a 3D printed container that we adapted to the coil shapes. This is a 20-channel coil in the Prisma. We had to plan the container in a way that it fits perfectly into the coil. We had to plan this is a container that was actually planned already in NIH. They gave us the protocols, we readapted to the Prisma, and we used their way of sucking out bubbles, which are something that postmortem you don't want to have. You don't want to have them anywhere, but postmortem is a problem because you use this brain, you put it in fumbling in this oily liquid, and then bubbles are there. And then all your efforts are crashing down with bubbles in the images, and you cannot see anything. So what we do is for 2 weeks, we suck out bubbles using a vacuum pump. So it's as simple as it is. We put this brain under vacuum. So, and this is what you see here. You see this valve where we put the vacuum thing. And after 2 weeks of shaking and debubbling, shaking and debubbling, this brain is ready for scanning. And then when we get the images, it's always Riccardo, he's reconstructing using the image again, protocol of NIH, they published it. Riccardo is making a 3D printed container of the brain. So each brain has its own container where you see these cuts. These cuts are 1 centimeter apart, and they're exactly what the brain slices, DMR slices are. So that our pathologists, but our collaborator pathologists can put the knife there. And they cut finally very precisely where we want. because pathologists are normally doing it a little bit like this. So they can cut obliquous and so on. So in this way, what we get are perfectly registered slides. We still have 2D registration in between, but the effect is really small. And what did we get? So what are we getting postmortem? This is an ongoing study, so we have so far scanned 5 brains, but we are aiming at 10. And we get also their specific quantitative contrasts. There we get the myelin water fraction, we get quantitative T1, so always MP2 range, we get QSM, all different kinds of QSM. We like it a lot now after the initial suspicion. And we also got an MTSAT that we have also in vivo. I didn't show you the studies using MTSAT. Anyway, these 4 maps that are acquired are then combined in a multi-parametric study, and you will see what Ricardo did. So you see the resolutions of these maps, and you see, you don't see it, but I can tell you there is a huge effort behind these acquisitions. And what did Ricardo do? So since we have the chance that we can use these slices, look at the images saying, hey, I'm interested in this lesion, this lesion, this lesion, tell the pathologist, we take the blocks, they analyze the blocks. We can also select the lesions that we want to analyze. And the pathologists are giving us back the response and telling us, hey, this is an active lesion. Active lesions look like that. They have many, many, many cells, ongoing demyelination, some remyelination rate. Here, the high remyelination rate is actually relative to the bioptic studies. So we don't see too much remyelination in active lesions, but this doesn't matter. Inactive lesions, they tell us there are these, again, a terminology which is not the same. These mixed active, inactive are exactly these rim lesions or paramagnetic rim lesions, whatever you want to call them. It's always the same stuff. So those are the bad lesions. And they tell us there are some remyelinated areas. These BICAs are actually oligodendrocytes that are re-differentiating, that are ready to go into the remyelination path. And this is a myelin stain. So this is the information we get from the pathologist. And then what did we do? Actually, what did Ricardo do? Ricardo looked at how these contrasts were behaving in these different type of lesions. And for this work, the ISMRM abstract, he looked at 34 white matter lesions. 11 were inactive, 11 had this rim, 6 active, and 6 remyelinated, or partly of them were remyelinated. Here I wanted to show you just the co-registration. It's not easy to do this level of co-registration, but I think Ricardo achieved quite a nice co-registration level there. And of course, he performed different statistical tests with multiple comparison correction, blah, blah, blah. We consider all these lesions as independent, despite the fact that some of them were from the single brains, because we will do better in the paper, but because in this case, pathologists are telling us that lesions are behaving independently in the single brain. So this has been reported. However, I think it will be much cleaner for a future paper that we also take into account the potential dependency of lesions from the same brain. This will come later on. But anyway, I wanted to show you the results. And those are the results. This is directly taken from the talk of Ricardo, so you see also the audio there. Ricardo showed that there was actually the expected trend in MTSAT and in T1. So MTSAT was decreasing progressively, going to from normal appearing tissue to inactive lesions and these rim lesions, which were the most destructive. And T1 was having the opposite behavior. So signifying a loss, most probably of myelin, because we are in white matter here, and a loss of myelin as well, so a loss of myelin and structure with the T1. Then myelin water fraction was behaving more or less like the MTSAT, a little bit more variable than MTSAT. And QSM is a mess. So QSM you cannot average because, of course, it's a complicated behavior of this map due to the interdependency, due to the double effect of the iron-related signal, which is a lot in these lesions. We have a lot of inflammation, and the orientation of myelin, which is changing if you have myelin damage. So this double effect makes it that it's very difficult to understand something from QSM, apart from active lesions, which have a high QSM point. So this is what we can say. And here I wanted to show you images, and you will see a lot of images in the next slides, and then we will go towards the end, because I'm speaking a lot and sometimes too fast. So inactive lesions, how do they look like? They look like this, a lot of damage. Voila. We show already low empty sat, low myelin water fraction, QSM a little bit hyper intense, and quantitative T1 very long, as expected. Active lesions, actually a little bit of reduction of empty sat, myelin water fraction a little bit, hyper intense, and quantitative T1 a little bit prolonged, not too much. And these famous rim lesions, again, a lot of destruction, expected. The rim in QSM, and quantitative T1, bit of prolonged quantitative T1. Again, yeah, this is very interesting. There is most probably a double effect of the iron accumulation and the tissue destruction. It's not very much prolonged, this T1, despite they're very much distracted, these lesions. And last, these remyelinated lesions, which I like so much. Here, remyelinated actually area. You see that the empty sat in certain parts is very similar to the surrounding tissue. It's actually much, sorry, no, this is a surprise, no. Much, it's getting higher compared to the center of the lesion, and same thing for the myelin water fraction. Quantitative T1 is also capturing this remyelinating process. And the QSM here actually has a lot of central veins. You see the veins there? Sorry. So it's appearing slightly hyperintense, but if you do a quantitative analysis, it's very similar to the surrounding tissue. Last, I wanted to show you some fancy images because Matthias really put a lot of thoughts. This is also another paper under revision. It's called URI, ultra high resolution images. So Matthias achieved these 200 microns isotropic at 3 Tesla in the whole brain. And for me, it's fascinating to see the anatomical structures there. I mean, those are examples of images, so take it as a imaging show. I don't want to show you any quantitation, but I want to show the little lesions in the thalamus, the different nuclei of the thalamus in these images. And then the super nice cerebellum, dentate nucleus. You see also the little lesions inside. And Matthias went down, because I'm always very demanding, he says he went down to 160 microns, and there you start seeing super tiny small cortical lesions, really tiny. I mean, you see them still at 240 microns, but I can assure you, as soon as you go to 300, 350, you don't see them anymore, of course, the small ones. You see the central veins inside the lesions, which is typical of NMS lesions because they develop along the vein. You see rims everywhere, also in the cortex. This is new. I mean, look at this. This is fascinating. This is blocking this the level of the cortex. Some rims are in the cortex. What the difference is between those two, we don't know, to be discovered. You see subpial lesions, also those, they are very difficult to see in vivo in patients, but we see them in the cell side. Look at this. Here you lose the structure of the cortex and you see the subpial lesion. You see a lot of them actually. You see? This is typical subpial lesion. And that's it for fancy images now. So this is another paper that we're still preparing the review, but hopefully it will be out quite soon. And I wanted to conclude again, coming back to our collaborations, because this is all collaborative study. But one important collaboration we have is, of course, with Lausanne. A lot of important collaborations. I wanted to show you a little bit where we are going here, because so far I showed you fancy stuff that let us perhaps understand new mechanisms of disease. This is at least what we think and what our hope is. But in the end, I mean, me from also being in the clinic 2 days per week, I always say, hey, we need to try to use these things for our patients already now, even if it's not ready, even if it's not, we have enough of information. Sorry. This is not what I want to show you. And here is the summary. This is actually a slide that Mary provided me, but it summarizes really well all our activities. You have seen that we have been collaborating for many years at different domains. What I'm I want to say here, I would not go into the details of these studies, of course, because it's a lot of work from Mary and Francesco and all the people in her lab. There was also a strong collaboration with the Siemens team through Mario Fartaya. All this work is, of course, coming from their teams. But the collaboration was really fruitful there because we could give the feedbacks that allow them to develop these tools to a certain extent. And now, hopefully, we can also try to really translate them in the clinic. And I wanted to show you two, which we are really trying to translate in the clinic, meaning use them clinically in single patients. That's what I'm trying to do. This is the tool for cortical lesion detection and white matter lesion detection. You see the collaboration here. It's also data sharing. So we have data from the SHU, from Basel. There is the implementation of the network from Mary's group. There is the outcome, which is good enough for us. So it's good enough for us to already start using it in single patients. There are some data that need to be manually corrected. This is normal, but this is an information that clinical patients can benefit to. So what we are trying to do is now to implement this in a clinical unit, which will be the clinical advanced MRI unit in Basel, where this tool may be used for will be used for single patients. And the same thing holds true for the rim lesion detection, these famous bad lesions. These are really interesting. There are clinical trials going on for those lesions. We can now not identify them in our patients in the hospital. And this study, in my opinion, is the necessary step to go forward and try to implement. This is a study where Mary, together with Herman and Francesco, of course, using data again from Lausanne and Basel, developed a tool to automatically detect these rim lesions so that we have a much easier life. I mean, we have masks, we can manually correct them. So it's a supporting tool, but it's rendering our life for single patients much easier. And this morning I was correcting a report with this. We are giving now for our patients numbers of those lesions, which are of course manually evaluated, but we are doing this. And so now to summarize, since I've spoken too much, of course I haven't spoken about all the collaborations we have. There are many more. And but I would like to summarize what I've said up to now. So I think that what I feel confident to say is that advanced MRI, both in vivo and post-mortem, is providing a fundamental contribution to the understanding of this complex pathology. Perhaps it's not even a pathology, perhaps it's a spectrum of diseases, perhaps it's a syndrome, call it as you want. But through these approaches, we could understand things. I mean, you've seen these 3 studies, and we have a lot of perspectives there. So we want to, for example, for the first study, we would like to use different type of diffusion models to better understand the microstructure of these lesions, and always in the framework of what is applicable, what is not to this type of data. But this is definitely a path I would like to exploit. The QSM, I mean, for me, is fascinating. These hyperintense lesions, they're a mine of information which we need to be able to understand. This personalized assessment of bad lesions in imaging, but also these biomarkers in the blood. The multi-contrast histopathology studies, there, what we are doing now, beyond the study of Ricardo, is to assess the different sensitivity and specificity to myelin and axonal density of these different quantitative contrasts. We have many studies that did only one part, only one sequence, only one assessment. Now we are trying to put the pieces of the puzzle together. And last but really not least is this advanced MRI in the clinic. I mean, the clinical unit is a reality. We will push for this to develop it. It's something that we were asked to do. And we will pursue definitely. So, and my goal is really to translate this type of tools that have been developed in these collaborations in the clinical practice for the single patients, and it's possible. So that's my positive message in the end. And here I wanted to, I mean, there are a lot of collaborators. You see it here. Just wanted to mention, I mentioned many times Mary. I mean, this is clear. It's a long collaboration. I also wanted to mention, for question of time, I didn't show any study that we did with Alessandro and Simona in Verona. We have a lot of studies going on there. of course, in the field of diffusion tractography. This is also fascinating work to better disentangle using quantitative MRI, diffusion, and so on. So this will be for the next CIBM seminar. But there are strong collaborations which have been developed over the years, over a long time, also with Alessandro. I don't remember now how many years it is that we are collaborating together. So I hope to continue and to be here perhaps in another 10 years to tell you more. And thanks a lot. And with this, I thank you all. And that's it.
Super. Thank you a lot, Cristina. Amazing talk. I really enjoy. I really like what you do. So your sense of rigor with this histopathology and pathologic study at the lesion level, I think this is super important, and the marriage that you do with the MRI and advanced MRI features, still with the foot on the ground for the patients. So it's really amazing. Thank you very much. We have few times for questions, but I would like the audience to not be shy, because we don't have time to lose. So do you have questions or comments for Cristina? I will try to get on the chat to see if there is in there.
No comments.
While people is thinking, I had a question for you. I probably we discussed already, but you're presenting here a lot of biomarkers, advanced biomarkers at the lesion level. And something that I do not see though is how it integrates the location or the relation of the lesions into that, because at a certain point you said we consider the lesions independently, but what's going on if the lesions are at a specific location? You said some locations were more dangerous, or if they are in the same white matter tract. Could this spatial information or whatever be included in your understanding of the patients?
Yeah, sure, definitely. So this is another part which, of course, we haven't addressed, and this needs to be taken into account. It's definitely different if you have a lesion or multiple lesions on the cortical spinal tract or in silent areas of the brain. I mean, for the purpose of understanding the nature of the lesions and of the pathology, this is not probably not very meaningful, but for the purpose of understanding the clinic, this is fundamental. So this is for sure another piece of the puzzle that needs to be added to. One is not enough. I mean, most probably two is also not enough. We have to have a comprehensive approach for these patients. That's why I tried to show you also the combination with the biomarker. It's for sure not with one single imaging technique that it will solve all the problems. We'll perhaps target a certain mechanism, like with these chronic active lesions or rim lesions, but the entire picture of the patients will require many more elements. Yeah.
I see in the chat that Katarina have a question. Oh, please go ahead, just unmute.
Yeah, I'm not, you can speak, I'm not for
I have one question because, I'm working also in inflammation and oxidative stress in the neurodegenerative disease. So my question is, because you're testing also different type of biomarker, did you test also the oxidative stress in the multiple sclerosis or not? Did you try to maybe input some medication, a part of inflammation, anti-inflammation medication to support? Because, you know, inflammation is like going together, you know, they are not apart. So you have both of them coexisting. So my question is, did you test some things, you know, to improve the condition of the patient, or still?
So you're speaking that wise. I'm totally biased because I'm imaging wise. So inflammation for us is iron, water. That's it. They have cells.
All the, you know, Haber-Weiss reaction, Fenton reaction, you know.
Sure. This is a
and everything going around.
Yeah. Clear. So this is a branch of research in MS for sure. I mean, there is also mitochondrial dysfunction.
Yeah,
there are drugs which have been tested. Sincerely speaking, they didn't show too much promise alone. So far, the drugs which were specifically targeting these paths. But this is.
They are rather anti-inflammatory than antioxidative. Yes. All of the drugs.
There's some of the, you know, if you block the initial path of inflammation, you block also the oxidation, which is one of the final.
Yeah, but you block the initial part when it's coming from the lymphocytes and then macrophages, but then
so then
one which is ongoing within the oligodendrocytes, which is going oligo in the neurons, which is
correct.
The microglia and astrocytes, you're not blocking.
No, well, at the present, not, but there is a lot to do. So that's why we need to do research. There are a lot of drugs which have been being tested for that. Again, I tell you that to my knowledge, this type of drugs didn't make it beyond the phase 1 trials so far. But of course, this is definitely something that we need to address. I mean, we are aware of this. I am not producing drugs and I am not developing drugs. So from my point of view, what I'm trying to do is to use these imaging approaches to get an insight in living patients and to understand a bit mechanisms that possibly we can target. If I don't know how we could target specifically that. I mean, of course, you could use PET with specific tracers to target this approach, and this requires a certain other infrastructure to do it. There is the costs which are associated to PET analysis. There is radiation which is associated. So it's a big. There are for sure groups working on that. So this is for sure. Yeah.
Thank you very much.
Thank you, Katarina, for your question. Cristina.
Ciao, Cristina.
Cristina, go ahead.
To see you. Very nice talk. I'm coming with another question which you haven't done yet. Do you hear me?
Not super well. Do you have a microphone, or can you be closer? Okay.
Sorry, do you hear me now?
Perfectly.
Good. So I'm coming, j'attaque avec une autre question que tu n'as pas fait. So with another thing that you haven't done. You have so you're talking about biomarkers. So you're looking to different biomarkers that we are looking in spectroscopy. But you have a model of inflammation. You have a lot of inflammation. And in spectroscopy, we do not have a marker of inflammation. So there was Miriam Chamay from UCSF, who was working a lot on multiple sclerosis in preclinical models. And she was doing DMP to try to find a marker of inflammation. She was talking about lactate, I think, in multiple sclerosis, and as a marker of inflammation. But I was wondering, do you plan to do some spectroscopy in the lesions and outside the lesions, or not at all? Because with the inflammation that happens and the lesions where you know that there is inflammation, you could see some spectroscopic changes which are related to the inflammation, and that would be quite novel because
So
inflammation,
the answer is easy. So first of all, I'm not doing everything. So I've showed you a lot of stuff. There's so much to do, fortunately. So the answer is easy. If I have a good collaborator, an expert in spectroscopy, definitely. I mean, I'm personally.
I'm one, huh?
Voila. So that was an indirect answer. No, definitely. This is very interesting. What is also very interesting for me with spectroscopy would be a bit the remyelination process. So this repairing process, which is also something that I think spectroscopy can help with. But you need the expert there, so Cristina.
Thank you. And again, it was such a big. It's such a big pleasure. It was not. It is such a big pleasure to see you and to hear you. Thank you very much.
Thank you, Cristina.
Thank you.
Is there other question? You can either use the chat or raise the hand.
Or just drop me an email if you're shy. Yeah. I'm always speaking a lot, so this is not unusual.
No, it was a very great talk. I had one more question for you, Cristina, if you don't mind, Pina. So just very quickly, you said it at the beginning, your graph of the progression in discrete time points is not correct. You know that we are in a continuum.
Yeah.
But how this continuum can be really modeled or tracked with MRI? Because MRI, you have it after an indication, right? So you as well as only having a sampling, I don't know how much you sample these patients in your follow-up. So
that's a very interesting question, actually, because I mean, we normally, clinically, we are lucky in Switzerland, we're monitoring these patients once per year, at least, even if you're stable.
Once per year?
Yeah. But, I mean, this is also an ongoing discussion that most probably to capture the subtle variation in size or even in repair that happen at a certain time, and you have the lesions, they're not synchronized, they're all different. You need a much more frequent sampling. So, and that's what we are trying to plan now, pilot studies, to have 1 scan per month, for example, without GADO. Or to have over 1 year, a couple of scans at the beginning of a treatment, just a very frequent sampling. Then in the middle of the treatment, then at the end, very frequent sampling, and see if we can better understand trajectories.
Okay, so I don't know, Pina, sorry, shall I give you the floor again? Sorry, I don't know what I have to do now.
I'm just going on with my news and events.
I see, she's on the chat.
So this way you all have the links to what is going on in CIBM, and thank you all for joining, and we are up to time. So thank you again, and look forward to the next one next month.